This is Biology for Grade 11, produced by Tigray Education Bureau, transmitted by Dimtsi Weyane Tigray National and FM radio satellites and social media, and also FM Maqala 104.4.
Hello students, welcome back to Biology lesson for grade 11. I am Gabrmara Mimana. Before we start our today's lesson, let's rehearse some points from our previous session about plasma membrane. All of you, as you can remember, plasma membrane or cell membrane is a membrane that surrounds and encloses a cell. It plays a crucial role in cell signaling and it controls what enters and leaves from the cell too. Because of that, cell membrane is the traffic of the cell. Okay students, our today's lesson is on how substance crosses the plasma membrane. From our today's lesson, you are expected to differentiate passive and active process, to list and explain the different type of passive and active processes, to explain the factors that affect the rate of diffusion. So, particles of any substance passes through the plasma membrane based on the following two processes. Those are passive process and active process. Passive process is a transport of substance through a plasma membrane without the expenditure of energy. Whereas, active process is the transport of substance through a plasma membrane by using an energy. Okay students, let's see the type of passive process and the active process. First, let's see what passive processes are. As we said before, passive process is the transport of substance through the plasma membrane without the expenditure of energy. It includes simple diffusion, facilitated diffusion, and osmosis. I hope students, from those three types of passive process, you identify one word, that is diffusion. So, what does that mean, diffusion? Okay students, diffusion is the movement of substance from higher concentration towards a lower concentration without the need of energy. For example, if your teacher sprays a perfume in one corner of the school or the class, then slowly the perfume diffuses to the whole class until it becomes equilibrium. So that is an example of diffusion. So let's see the three types of passive process one by one. The first one is simple diffusion. Simple diffusion is the movement of substance from higher concentration to lower concentration without the expenditure of energy. It tends to distribute molecules uniformly in random motion from higher to lower until it becomes equilibrium at both sides of the plasma membrane. To pass the plasma membrane by simple diffusion, particles must be small, lipid-soluble, and non-charged. However, particles like ions, sugars, amino acids, and any large particles cannot pass or cross the plasma membrane by simple diffusion. They need another aids. The rate of diffusion can be influenced or affected by three main factors that are concentration gradient, thickness of the membrane, and surface area of the membrane. So let's see their effect on the rate of diffusion. Concentration gradient. As the concentration difference in the two sides of the plasma membrane increases, the rate of diffusion also increases. Thickness of the membrane. The thicker the membrane or the barrier, the lower the rate of diffusion. Surface area of the membrane. The larger the surface area, the faster the rate of diffusion. But in addition with those, the rate of diffusion can be also affected by temperature. Rate of diffusion is equals to surface area of a membrane times concentration difference over diffusion distance or thickness of the membrane. This equation, also called Fick's law of diffusion. Based on this equation, the rate of diffusion is directly proportional to the surface area of the membrane and the concentration difference, whereas it is inversely proportional to the diffusion distance or the thickness of the membrane. Okay, students, I hope you are attentively following my presentation. If so, let me ask you one question. What kind of particles can cross the plasma membrane by simple diffusion? What kind of particles can cross the plasma membrane by simple diffusion? Alright, students. If we say particles that are small, non-charged and lipid soluble, you are right. The second passive process is facilitated diffusion. Facilitated diffusion is the movement of substance from higher concentration to lower concentration through an ion pore or carrier protein. Facilitated diffusion has the following characteristics. 1. It is specific. Only a certain molecules can pass through the channel or the carrier proteins. It is also passive. That means particles move from higher concentration to lower concentration without the need of energy. The rate of facilitated diffusion can be influenced by the number of carrier or channel proteins present on the plasma membrane, the concentration gradient, the thickness of the membrane, and the surface area of the membrane. Okay, students, let me ask you one question. What is the difference between simple and facilitated diffusion? What is the difference between simple and facilitated diffusion? Alright, if we say simple diffusion is the movement of substance from higher concentration to lower concentration without the need of any aid, whereas facilitated diffusion is the transport of substance from higher concentration to lower concentration through an ion pore or a carrier protein, you are right. Okay, students, let's proceed to the third passive process, that is osmosis. Osmosis is the movement of water molecule from higher water potential or less negative to a lower water potential or more negative through permeable membrane. It is also called the diffusion of water, and osmosis is a special kind of diffusion. Water potential is the concentration of water molecules. Water potential value in a solution is always negative because pure water potential is equals to 0 Pascal. Okay, students, try to answer this multiple choice question. What will happen to the water potential if the amount of solid in a solution is increased? A. It becomes more negative. B. It becomes less negative. C. It becomes 0. D. It becomes positive. I will repeat it, the question. What will happen to the water potential if the amount of solid in a solution is increased? A. It becomes more negative. B. It becomes less negative. C. It becomes 0. D. It becomes positive. Fantastic, students. If we say A, you are right. That means as the concentration of the solid increases in the solution, the water potential value becomes more negative. Now, let's see what factors affect the rate of osmosis. Rate of osmosis is influenced by surface area of the membrane, difference in water potential, and distance the molecule must travel, or thickness of the membrane. Okay, students. Osmosis in living cells exists in 3 different types of solution. For example, our cells are based in a blood, so a blood is a solution. Then, a cell can be placed in 3 solutions: isotonic solution, hypertonic solution, and hypotonic solution. So let's see them one by one. Number 1, isotonic solution. Iso means equal. Here, the water potential inside the cell and the water potential outside the cell in the solutions are the same. Because of that, there is no water movement into or out of the cell. So, any cell immersed in isotonic solution remains as it is. The second solution in which a cell can be placed is hypertonic solution. In this solution, water potential outside of the cell is lower than water potential inside the cell. So, water moves from the cell out to the solution. Then, the cell loses its water and gets shrink. If the cell that gets shrinked is an animal cell, we call it crenation. Whereas, if the cell that gets shrinked is a plant cell, we call it flaccid. However, here, if a plant cell shrinks too much and lost its contact of cell membrane with its cell wall, or if the cell membrane is detached from its cell wall, we call it plasmolyzed. The third solution is hypotonic solution. Here, the water potential in the solution that is outside of the cell is higher than inside the cell. So, water moves from the outside of the solution into the cell. So, if the cell is an animal cell, it takes water, swell, and finally burst because it has no cell wall. We call this condition hemolyzed. Whereas, if the cell that placed in hypotonic solution is a plant cell, it takes water, swell, and becomes turgid. It does not burst because it has a cell wall that exerts a wall pressure against the turgor pressure of the water. This turgidity is important in supporting young non-woody plant stem, or this turgidity in young non-woody plants is used as a skeleton in animals. Okay, students, let's try to answer the following questions. Question number 1. What is plasmolysis? What is plasmolysis? Great job, students. If we say plasmolysis is a process of detaching the cell membranes of a plant cell from its cell wall because of the loss of too much of water when it immersed in hypertonic solution. Question number 2. At what condition animal cells hemolyzed? At what condition animal cells hemolyzed? If we say, when an animal cell is immersed in hypotonic solution, it becomes swell and final burst. So this process is hemolyzed. You are right. Okay, students. Let's proceed to the second main process in which substance crosses the plasma membrane. That is active process. Active process is the process in which substance transports through the plasma membrane by using an energy. This active process includes active transports, endocytosis, and exocytosis. Let's see them one by one. Active transport is the movement of substance against a concentration gradient, or from low concentration to high concentration using an energy. For example, absorption of minerals by plant roots from the soil is by active transport. The second active process is endocytosis. Endocytosis is the process of engulfing large particles by the formation of vessels on plasma membrane. At that time, it uses an energy from ATP. There are 3 types of endocytosis. The first one is phagocytosis, the second one is pinocytosis, and the third one is receptor-mediated endocytosis. Phagocytosis or cell eating. It involves the creation of pseudopoda to enclose large particles or even the whole cell. It is used by protozoa and several classes of white blood cells to ingest solid particles such as bacterium. Pinocytosis or cell drinking. It involves the ingestion of smaller particles and does not require formation of large pseudopoda. It differs from phagocytosis only in scale. Cell takes in dissolved materials or liquids by pinocytosis. The third endocytosis is receptor-mediated endocytosis. Here, the membrane enfolds to form vessel only in regions where the particles have bound to a specific receptor. For example, cholesterol in the bloodstream is taken up by animal cells by receptor-mediated endocytosis. The third active process is exocytosis. Here, substances are moved from the inside to the outside of the cell in reverse of endocytosis. For example, enzymes and hormones are secreted by exocytosis to the bloodstream. Okay, students. From the information that you gain from the active process, try to answer the following question. What is the difference between phagocytosis and pinocytosis? What is the difference between phagocytosis and pinocytosis? Okay, students. If we say phagocytosis is the ingestion of large particles or even the whole cell by the formation of pseudopoda, whereas pinocytosis is the ingestion of smaller particles without the need of the formation of large pseudopoda. You are right. Okay, students, let's wind up our today's lesson. Substance crosses the plasma membrane in 2 main ways. a passive process and an active process. A passive process is the transport of substance without the expenditure of energy. It includes simple diffusion, facilitated diffusion, and osmosis. However, active process is the transport of substance through a plasma membrane by using an energy from ATP. It includes active transports, endocytosis, and exocytosis. This is all about our today's lesson. Next time, we will see on the different cellular organelles. Till that time, goodbye.